let's get back to work. So, during the break, Brittany and I labeled one of the coronal sections. So your handout, page 1, this is through the frontal lobe anterior to the thalamus. Your handout should have labeled something like this, especially the structures of the putamen, the caudate, and the globus pallidus. So that's new information. But remember, I think that it's important to print out another blank and label it to learn it. So this is not labeling everything. Let's see, how many labels do we get? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 labels. Questions? We didn't label any lobes, so temporal lobe, so there's 12. Questions about this? So I think, you could do the same thing with this one, which is now wrong because this is the thalamus here. Okay, so don't pay attention to those labels because they're only right here. Any questions? Let's talk for a few minutes about the hippocampus and what's going on in your brain right now as you form new memory. So here is the hippocampus. This is a section, this is a cartoon temporal lobe, cutting away of the temporal lobe so that we get to the medial temporal lobe. So we're seeing the hippocampus having trimmed away part of the temporal lobe till we get to the middle of it, and there's the hippocampus in the medial temporal lobe. It actually drops around here if we go deeper there. Hippocampus just is Greek for seahorse. And you can see why some neuroanatomists called the hippocampus. Looks like a little seahorse in the brain. Here it is in a coronal section. Medial temporal lobe. So in one section right there. But it goes back and comes out here, goes back there. So a little sausage-shaped thing. And if you cut it in the right place, there it is. The hippocampus is a remarkable structure because it has a very well-defined laminar structure. Well, I'm not going to ask you to learn the names of the different parts of the hippocampus. I do, and you don't have to. I just want to show you something beautiful. So here's a hippocampus where the neuron, so this isn't colorized. This is actually, each neuron was labeled with a different mixture of fluorescent molecules. And so this is a mouse hippocampus. It's not a human hippocampus. This is a mouse hippocampus where different neurons were labeled with different fluorescent molecules. So you can see the different ones because each one is slightly different color. It's got the name of the animal is the brain bow, like rainbow, only brain bow mouse. So you can get these mice and they make colored neurons. But you can see this well-organized laminar structure. Cell bodies, axons making synapses. Cell bodies, axons making synapses. Here's a cartoon of the same thing, right? So, sorry, we'll just leave it off now. I mean, this is good. This is good. So you can see cell bodies, axons making synapses, cell bodies, axons making synapses. The hippocampus is where a phenomenon, now we're going to transition to physiology again. There is a phenomenon that was discovered in the hippocampus, but turns out is true all over your brain, called long-term potentiation and long-term depression. Depression is not, we're not talking about clinical depression, we're talking about neuroscience depression, neurobiological depression. Long-term potentiation is an increase in synaptic strength that lasts for a long time, hours to days. Long-term depression is the reverse of that, reversing that process. And here is what long-term potentiation looked like when it was discovered. Let's get oriented. Here is a synaptic response, typo error in this review article, as a function of time. So this is the amplitude of EPSPs, frankly. This is just EPSP amplitude in a section of the hippocampus. And they normalize it to 100%. So they get a baseline, and they say, okay, that's baseline. We're just going to call that 100%. That's baseline. Then they stimulated those neurons with a burst of action potentials. Like a real fast action, a fast train of action potentials. And they get a potentiation. They knew that before. They knew that there would be synaptic potentiation if they stimulated those synapses fast. But what they discovered was after this potentiation, which is short term, only lasts a few minutes, it settles down to a new baseline that is higher than the original baseline. Everything about this stimulus is the same as this stimulus, but the response is bigger. It's been potentiated, and it lasts for a long time. This is just showing 1 hour, but it can last for 6 hours, and it can last for 6 days. The mechanisms for a 1 hour long term and the mechanisms for a 1 day long term are different. We're going to focus on the mechanism for this form of long-term potentiation. I love this story because it uses your knowledge and it tells you how you are learning. So I love this story because it's interesting, it's fascinating, you are learning things because of this story, and it uses the physiology that you've already learned. So let's tell this story. And I don't think I gave you, did I give you this story? Yes, I gave you the story nice and big so you can actually see it. Let's tell this story using your neurophysiological knowledge. So here's the presynaptic neuron that was stimulated very fast. It released a neurotransmitter, an excitatory neurotransmitter, the excitatory neurotransmitter glutamate. Okay, so I just skipped a story. Action potentials, release of glutamate. You should be thinking in your head the story that connects action potentials to the release of glutamate. You could tell me that story. We're not going to do it now because it's review, but you should be able to, I could tell it real fast, but you should know, things like voltage-gated calcium channels, calcium influx, calcium binding to synaptotagmin, conformational change of the snares, fusion of vesicles, diffusion of neurotransmitter. I just gave it to you, but too fast for you to write down. But the point is, you have learned a story, and sometimes I skip over it, But you should not skip over it. If I say, tell me this whole story, you want to tell me that part of it. Glutamate is the neurotransmitter at this synapse. Excitatory. There are two types, let's see. Two types of glutamate receptors. I am not going to ask you to memorize, just like I didn't ask you to memorize the names of different muscarinic receptors, I am not going to ask you to memorize the names of different glutamate receptors. Okay? One of them has a name called AMPA, and the other has a name called NMDA. Right? They have different names. And I'm happy to tell you why they have those weird names, but let's not get into it. Glutamate activates AMPA receptors. AMPA receptors, like acetylcholine receptors, are sodium and potassium permeable. So are these excitatory? These are ionotropic receptors that are sodium and potassium permeable. Do they cause a depolarization or a hyperpolarization of this dendrite, the postsynaptic membrane? Depolarization. Sodium influx is more overwhelms the potassium efflux, and it causes a depolarization, just like the acetylcholine receptor did in your problem set. Depolarization. The NMDA also activates the other type of receptor, but now we've got an interesting phenomenon. If this membrane is not very depolarized, just a small depolarization, this receptor is blocked by magnesium. So this is a detail you don't need to know. But this receptor, even though it's binding glutamate, it's blocked. But if there's been a train of action potentials that has depolarized this postsynaptic cell a lot, then this area is depolarized and it kicks the magnesium off. Now this receptor is activated and unblocked. Now this receptor, sodium can flow in, causing further depolarization, but here's the twist. This receptor is also permeable to calcium. It's not a voltage-gated calcium channel, it's a ligand-gated receptor that is permeable to sodium and to calcium, and to potassium for that matter. But calcium flows in. So now we've got a calcium signal. So you might want to say, okay, calcium goes and binds to the IP3 receptors, and that's not the story here. The story here is that calcium is coming in from the outside through this channel and activates calcium binding protein, an effector, calmodulin. This was a French review, so it's calmodulin. Calcium binds to calmodulin and activates it. That activates kinases. Kinases activate cascades. This kinase phosphorylates these receptors and makes more of them and makes them more active. So what do? With a single action potential, slow, all we got is normal neurotransmission. With a fast pulsatome, we got this channel unblocked, got calcium, got a story of kinases that put more receptors into this membrane. More receptors, bigger EPSP. More receptors, bigger EPSP. That lasts for hours. Here is a change that activates genes. Now we got a story that lasts for maybe the rest of your life. I'm hoping that some of the things we talked about today have activated this pathway. So here's the story in words. Here's the story in picture. I could ask you a story. I could ask you to tell me a piece of the story. Especially the pieces that reinforce the generic stories that we've been talking about for this past 2 weeks. Questions about long-term potentiation? A similar thing happens in your reward pathway. So I'm not going to go into it. I'm going to resist. I'd love to tell this story too, because it explains the reward pathway, and it explains why some drugs, like cocaine, hyperactivate the reward pathway and cause addiction. So I could ask you, okay, so if you know that cocaine, and here's the story that you could tell now, if I give you one bit of information, cocaine is an inhibitor of the transporter that transports dopamine from this synapse back into the presynaptic cells. Okay, I could quiz you. In order for dopamine to be reuptake by the dopamine transporter, what is co-transported? Sodium. It's a sodium dopamine co-transporter base. In order for dopamine to be pumped into vesicles, which aren't shown here, we need the proton pumps, and we need the proton gradient, and we need another transporter, the proton dopamine antiporter. So that whole story that is not shown here could be part of your knowledge, using your knowledge. Dopamine, G stories that you should be able to tell me, the GS story, the GI story, the GQ story. Remember, those of you who lost a lot of points on the problem set, on the question about the GQ story, you should use my answer for the problem set on Moodle to see the level of detail that I expect you to know. Binding, conformational change of the G protein, GDP dissociation, GTP association, G protein activating phospholipase C. A lot of you sort of skipped that whole part of the story and lost points. Don't do that on the exam. Put that story in your head for GQ, for GS, and for GI. But here's the GS story. GS, protein kinase A, genes, change, addiction to cocaine. Fascinating. So, I am of two minds about what to do next.
I just have a question on this. So this, in the addiction here's the stuff, so how is that affecting the
Inhibiting, so cocaine inhibits dopamine transport. More dopamine in the synapse for more time. More activation of pathway. More activation of genes. Change in the response. Change in behavior. Because of too much dopamine in this synapse. That was a good question. So, like I said, I'm of two minds of what to do now. What we have coming up is a series of anatomical figures that you could label. Superior view, right? Frontal lobe, occipital lobe, longitudinal fissure. So, lateral view, superior view, midline sagittal view, coronal view, different coronal view, and a bunch of clickers. So in my planning, I had planned to send you to breakout rooms at this point and work on these problems, these labeling problems together, teaching each other what you know and asking questions, because sometimes asking a question is as informative as telling somebody what you know or don't know, clickers. So, but I know that, so the alternative, the reason I'm on two minds is just to say, you have the resources needed to review anatomy, physiology, the series of clickers. So let me get a show of hands. No, I don't want to do it that way. What I'm going to do is open up the breakout room Zoom. If you want to work, so this is optional, if you want to work with other students and either help them, which is helping yourself, because there's no better way to learn than to teach. You just have to trust me on this. There is no better way to learn this material than to teach it to somebody else. So those of you who know it will know it better if you explain it to your colleagues, your friends, your peers, your classmates. If you don't know it, then you'll hear it again from a different voice than mine, which is stimulatory and may help you learn. So I'm going to form the breakout room. You go, if you want to, you go to that breakout session. Remember on Moodle, it's not the same one as the lecture, it's the breakout Zoom. And then I'll break you out into reasonable sizes. Those of you who show up in 10 minutes or so, I'll break you out into the breakout room. I will be monitoring, so if you have a question, you simply call me to your breakout room and I will answer your question. So I will be available to help with your breakout room. But it's optional. And we will not come back here to go over it together. You'll go over it together in your breakout. Or you'll go over it again on your own, if that's what you prefer to do.